In this lesson, we're going to contrast transmembrane and intracellular receptors. So first, transmembrane receptors, as their name indicates, span the membrane. So if at the top here, this is the outside of the cell and this is the inside of the cell, a transmembrane receptor spans the membrane like this and has a region on the outside that will bind the ligand, which I'm going to draw here in red. The ligand stays outside of the cell, and then a signal transduction pathway or a signaling cascade carries the message through the inside of the cell. The ligand stays on the outside. Another kind of transmembrane receptor is an ion channel. So drawn in cross-section here, there's an open pore here in the middle, or a pore that can be opened, and when the ligand, here drawn in red, binds, it opens this ion channel and allows ions which wouldn't normally be able to flow through the hydrophobic cell membrane to flow in or out of the cell through this pore. So an example would be sodium flowing into the cell. We'll look at these different types of transmembrane receptors in more detail in the next lesson. But in either case, the ligands don't cross the cell membrane. In contrast, with intracellular receptors, as the name indicates, the receptor is inside the cell, either in the nucleus or the cytoplasm. So this might be the receptor here, and the ligand, because it is small and hydrophobic in this case, can actually cross through the membrane. So here's the ligand going from the outside of the cell to the inside of the cell, and it can bind to the receptor inside of the cell. So in the case of intracellular receptors, the ligands do cross the cell membrane. Just a note about the structure of transmembrane receptors in terms of their amino acids. In order to span the membrane like this, the parts that stick into the watery aqueous solution inside and outside the cell, those are going to have hydrophilic amino acids predominant. So hydrophilic amino acids on the part that are in the aqueous solution outside and inside the cell, whereas the parts in the membrane itself, in the phospholipid bilayer, that's going to have hydrophobic amino acids to help it embed in the membrane. So transmembrane receptors are embedded in the membrane, intracellular receptors are in the cytoplasm or the nucleus. If we carry on thinking about the fact that the ligands don't cross the cell membrane in the transmembrane receptor case, the ligands can be large or they can be hydrophilic. Again, they're not able to cross the membrane, and that includes things like protein hormones such as insulin or oxytocin, or neurotransmitters made from amino acids like serotonin or dopamine. In the case of intracellular receptors, again, they're crossing the cell membrane, so they must be small and hydrophobic. And an important group of these are the hormones made from steroids like testosterone, estradiol, which is one of a class of hormones called estrogens, and progesterone. As we saw in the diagram above, in the transmembrane case, the original signal, the original chemical messenger, stays outside the cell, but it triggers a cascade of intracellular effects via second messenger systems. So we use the term second messenger to refer to the molecules that are carrying the signal in the signal transduction pathway. The first message is the original ligand that stays outside of the cell. The second messengers are the ones in the signaling cascade inside the cell. As I mentioned in the intracellular receptor case, the receptors can be in the cytoplasm or the nucleus, as opposed to being embedded in the membrane, as is the case with the transmembrane receptors. And as we saw above, an alternative, instead of triggering second messenger systems in the transmembrane case, is that they can open an ion channel, and that is part of the same protein as the receptor. I used the example of sodium moving into the cell above. In contrast, in some cases in the intracellular receptor case, the ligand receptor complex, if it's in the nucleus, may even bind directly to DNA and act as a transcription factor, regulating gene expression, meaning switching genes on and off and regulating whether they are expressed to proteins or not. So that is a compare and contrast between transmembrane receptors and intracellular receptors, and in the next lesson we'll go into more detail into the different kinds of transmembrane receptors.